Hello, and welcome to Neuromyelitis Optica. Are we closer to a rational therapy? My name is Bruce Cree. I'm an associate professor of clinical neurology at the University of California, San Francisco. I have received personal compensation for consulting from MedImmune regarding the design of a clinical trial in neuromyelitis optica. What is neuromyelitis optica, or NMO? NMO is a syndrome of aggressive inflammatory demyelination that affects the optic nerves and spinal cord and is also known as Devic's disease. NMO has been associated with infections and collagen vascular diseases, but has an idiopathic form that is considered to be a form of central nervous system demyelinating disease. Definitions vary, but experience from modern case series indicate that NMO is characterized by recurrent attacks of optic neuritis and acute transverse myelitis, multisegmental involvement of the spinal cord at least 3 vertebral segments in length, is often associated with a normal brain MRI, and many patients test seropositive for antibodies against aquaporin 4, a water channel that is expressed throughout the central nervous system as well as within the gut and muscle. Currently, multiple medications are used empirically for prevention of NMO relapses, and these immune suppressants include azathioprine, mycophenolate mofetil, rituximab, daily prednisone, and mitoxantrone. None of these treatments is proved to be effective because randomized controlled trials have not been conducted with any of these medications, and the data supporting their use is based on observational studies that are open label. In these studies, patients are treated with the medication, and the number of relapses are recorded either before initiation of treatment or after initiation of treatment. And so a decline in relapse frequency has been interpreted as being consistent with the treatment effect. The question is, are these case series adequate for definitively determining efficacy? The decline of relapse frequency observed in these open-label case series could be independent from treatment. The natural history of neuromyelitis optica is for attacks to cluster together, often around disease onset, and so therefore a period of time after that cluster of attacks is often a period of time of quiescence, and this might be occurring independently from treatment. Regression to the mean is also a phenomena that will cause overestimation of the treatment effect. And this means that since a treatment is initiated as a consequence of an attack, the immediately period of time following that attack will often be associated without an attack, and therefore the phenomena of regression to the mean results in an overestimation of a proposed treatment effect. Thus, to make meaningful claims regarding efficacy, a parallel group is always needed, either an alternate treatment or no treatment. We also know that in these case series, treatment selection is influenced by both known and unknown biases, and those can be introduced by both clinicians and patients. Matching can control for some of the known confounders, but unknown confounders can only be accounted for by randomization. And in all these case series, data acquisition was usually retrospective and unblinded and therefore subject to bias, although one of the trials with eculizumab was in fact a prospective study. Lastly, all the case series that have been reported in NMO have a relatively small sample size and therefore might not be representative. Some examples of observational studies in which randomized trials did not support accepted medical benefits include hormone replacement therapy in postmenopausal women, use of mycophenolate mofetil in myasthenia gravis, embryonic substantia nigra transplantation for Parkinson's disease, and use of donepezil for memory impairment in MS. In all these examples, observational studies supported use of these interventions that ultimately were proven to be not effective when randomized controlled trials were performed. So the question is there a case for equipoise in NMO treatment? Case series do provide some suggestive evidence of efficacy, but proof really does require randomized controlled trials with validated endpoints. And so the question is, what about using an active comparator for NMO clinical trials? There are currently no approved therapies, and therefore comparisons made against a treatment that is actually harmful could result in assigning benefit to a treatment that had no effect and only appeared to be beneficial because of the alternate treatment arm causing harm. And an example of this is the CAST study, which was a cardiac arrhythmia suppression trial. And in that study, both flecainide and encainide actually did worse than placebo. So there is a case for no treatment, and placebo control, of course, is no treatment. And this can provide unequivocal proof of efficacy. Some of the advantages of a placebo-controlled study include that the number of subjects participating in a placebo-controlled trial will be smaller than the number in an active comparator trial. And also, the number of events needed to prove efficacy will be smaller for a placebo-controlled trial. All clinical trials, of course, require medically relevant events in order to determine differences between two treatment groups. And so it comes down to how does one balance the risks of placebo in a randomized controlled trial in NMO, knowing that the events that occur in NMO are often clinically significant events such as transverse myelitis or optic neuritis that can result in a significant amount of disability for patients. Several design considerations are important to think about when considering use of placebo, especially in an NMO trial or really in any rare neurological disease. And the details of the study design become all important for assessing an individual risk. Unequal treatment allocation can help reduce the overall exposure to placebo, and time to first event study designs also will limit duration of exposure to placebo, as well as to determine what might occur after a particular attack has occurred. Use of rescue therapy and inclusion of rescue therapy as part of the protocol design is important. And lastly, limiting the total duration of placebo exposure can help limit the amount of time that patients might be exposed to placebo. An incentive for many patients is open label extension, meaning that if the patient has met that first clinical event, such as experiencing a relapse, or has made it to the end of the placebo-controlled portion of the study, they can be rolled over into an active treatment extension. And that's of lot of interest to many patients who would consider the risk of placebo in balance with the opportunity to receive a new medication for an extended period of time. Lastly, the use of placebo reduces potential treatment-related harm due to unexpected off-target effects. And this is particularly true for new medications that have not been investigated extensively in the setting of clinical trials in the disease under study. So when might the use of a placebo control be considered ethical? And there's a certain logic of clinical purpose here that has been previously studied. This is the case when there is no standard therapy, and the fact that there are multiple empiric therapies in NMO suggests that there is in fact no standard therapy. When standard therapy is no better than placebo, and therefore there is scientific equipoise, this certainly seems to apply to all empiric therapies since none of these have been proven rigorously. And then when there is doubt regarding the net therapeutic advantage of standard therapy, meaning an uneven risk-to-benefit ratio, and we're going to talk about this a little bit more, certain therapies currently used carry real risks to our patients. Standard therapy may not be available, and an example of this is rituximab, which in certain areas of the world is available for treatment of neuromyelitis optica, but in other areas of the world is not. And lastly, when standard therapy is placebo, and that is not the case for NMO. Equipoise has two requirements. One is genuine uncertainty regarding the relative scientific and clinical merits of each treatment arm. And the second requirement is that each treatment arm be consistent with best medical practice. And it's really this second arm that has been the crux of the debate regarding use of placebo in NMO trials. The question here is, can no treatment be considered competent medical treatment? And this really comes down to the risks and benefits of active treatment. And as we consider some of the active treatments that are used empirically in NMO, one has to realize that these active treatments carry real bona fide risks. For azathioprine, in a study of 99 NMO patients, 3 developed lymphoma, and lymphoma is a well-recognized risk of azathioprine treatment. And the question, of course, is, in a clinical trial, is this an accepted risk for an otherwise unproven therapy, and should this therapy be generally used in clinical practice? Perhaps other therapies such as mycophenolate mofetil might be safer. Well, right from the prescriber information, polyoma virus-associated nephropathy, JC virus-associated progressive multifocal leukoencephalopathy, cytomegalovirus infections, as well as reactivation of hepatitis B and hepatitis C have been reported, as well as lymphoproliferative disease or lymphoma occurring in a small percentage of patients treated with mycophenolate mofetil. So in clinical practice, how does one explain the risk of PML or lymphoma for an unproven treatment? And are NMO patients routinely screened for JC virus serology before starting and during MMF therapy? And are these risks even applicable to NMO patients, or do these risks apply only to transplant patients? And there are no simple answers for any of these questions. Currently, there is a clear need for clinical trials in NMO. There are no plans right now to pursue clinical trials with azathioprine, mycophenolate mofetil, or rituximab, or its humanized version, ocrelizumab, in NMO. Randomized controlled trials are difficult to organize and are unlikely to be funded by national research organizations. The cost of clinical trials is really quite high, and the relative scarcity of NMO has made this illness unattractive for taxpayer-sponsored research. And until recently, the pharmaceutical industry has been largely disinterested in NMO. The regulatory environment has gradually changed, and regulatory agencies in the United States, Europe, and Japan recognize that NMO is a distinct disease entity and is not simply a subtype of multiple sclerosis. The availability of the anti-aquaporin 4 antibody, or also known as NMO IgG serology, has changed the definition of NMO from Devic's disease to Neuromyelitis Optica spectrum disorder, and although rare, this disorder is probably much more common than once thought. Proven therapies are still not available, and placebo-controlled trials are now accepted by multiple regulatory agencies as well as by local and central IRBs. So there's a pressing need for international collaborative efforts to create cohorts for NMO patients to understand therapeutic response. And randomized controlled trials are really the best way to assemble such cohorts. Randomized controlled trials have a clear primary endpoint to determine whether a given treatment is effective or not. Randomized controlled trials provide the basis for regulatory approval, and regulatory approval subsequently provides the basis for payer support. And payer support, of course, is the mechanism that ultimately enables providers to prescribe medications to their patients. So there's a lot to gain from randomized controlled trials. Approved, readily available therapies whose side effect profiles are well documented in NMO will enable providers to accurately counsel their patients regarding treatment options. Clinical trials can form the basis of long-lasting collaborations, including long-term follow-up studies and studies of comparative efficacy. And clinical trials provide an unparalleled resource for acquisition of biological samples that could lead to subsequent breakthroughs in understanding disease pathogenesis. And this applies to NMO with respect to T-cell biology, B-cell biology, and the role of the microbiome in NMO. And there are multiple other examples from other disease states. Just one I mentioned here, which is the impact of vitamin D levels on MS disease activity that were identified in the benefit study of interferon beta-1b in clinically isolated syndromes suggestive of multiple sclerosis. The NMO clinical trial landscape is really quite interesting right now, and I'm going to mention 3 different products which are in development and actively recruiting patients for study. The first is eculizumab. Eculizumab is a monoclonal antibody that neutralizes complement protein C5. The entry criteria are for seropositive NMO patients who are over the age of 18 and have some ability to ambulate but might be using a wheelchair at least part of the time. And this study really is focusing on highly active NMO, and the requirement is that patients have had 2 relapses in the last 12 months or 3 relapses in the last 24 months. This study uses 2-to-1 randomization and is a time to first event design and uses a placebo control for eculizumab until the end of the study, which could be up to 2 years. This study does allow for ongoing use of azathioprine or mycophenolate mofetil, but is restricted to exposure to rituximab with last treatment with at least 3 months prior to entry into the study. Currently, eculizumab is indicated for paroxysmal nocturnal hemoglobinuria and is associated with known serious adverse events, including meningococcal sepsis and aseptic meningitis. Treatment is every 2 weeks and will be long-term, and in some ways is similar to natalizumab treatment in multiple sclerosis. Natalizumab is FDA approved for treatment of MS. It binds to lymphocytes and prevents lymphocytes from gaining egress into the central nervous system, and so it doesn't really alter the fundamental basic immunology of the disease. And I think of eculizumab as having a similar role in NMO, where complement fixation is needed in order to cause astroglial injury in the setting of anti-aquaporin 4 antibodies. Another clinical trial is looking at SA237, a monoclonal antibody directed against the IL-6 receptor. And this is similar to another product called tocilizumab. The group of patients targeted have to have NMO spectrum disorder between the ages of 18 to 74, and like the eculizumab trial, uses 2-to-1 randomization and a time to first time to first event design. This is also placebo-controlled until the end of the study. And in the United States, other immune suppressant therapies are prohibited, but these are allowed elsewhere in the world. SA237 is administered by IV infusion every 4 weeks. A similar compound called tocilizumab is indicated for treatment in rheumatoid arthritis and juvenile rheumatoid arthritis, and is associated with known serious adverse events, including infections leading to hospitalization or death, with tuberculosis, bacterial infections, invasive fungal, viral, or other opportunistic infections. And I think of SA237 and tocilizumab as being somewhat akin to daclizumab as a type of immune modulating treatment, but still one that does not alter the fundamental immunology of the disease. The third trial I will discuss is one with MEDI551, a monoclonal antibody directed against CD19. And this is an antibody that depletes B cells, pre-B cells, and plasmablasts. This study targets aquaporin 4 seropositive NMO spectrum disorder patients, but also allows seronegative patients who meet more stringent criteria for definitive NMO, and allows patients to be recruited over the age of 18 with no pre-specified upper age limit, and allows patients to be recruited up to an EDSS of 8 in certain cases, meaning that those patients might be bedbound at the time of recruitment. Like the other studies, it uses a time to relapse design, but limits the duration of exposure to a maximum of 6 months and uses unequal randomization with a 3-to-1 ratio. Other immune suppressant medications are prohibited in this study, and it is administered by IV infusion every 6 months. It incorporates an open-label extension for up to 3 years after the first event or after completion of 6 months of the placebo-controlled phase of the study. This is the first clinical trial with MEDI551 for any indication, and interestingly, this product has the potential to alter the fundamental immunology in NMO. So, it's estimated that NMO affects somewhere between 1 in 50,000 to 1 in 100,000 persons. And given that there are about 3 million people living in the United States today, therefore there must be between 3,000 and 6,000 persons affected by NMO in the United States. Globally, NMO could affect as many as 140,000 people, and therefore it has a similar population prevalence to other conditions that are considered to be rare, such as ALS. And for these trials, we need the participation of approximately 500 persons with NMO. So it's going to be a challenge identifying all of these individuals because that represents a significant proportion of patients in the United States who would participate in such studies. And therefore, all these trials have to have a global reach. I believe that we have the opportunity to have a profound impact on the lives of our NMO patients through systematic investigation of these new products. Assuming that these trials are successful, the contributions to the study subjects and the investigators will directly benefit the lives of thousands of others affected by NMO. And choosing to participate in these studies is really siding with the path of medical progress, regardless of whether these clinical trials are ultimately successful with respect to proving therapeutic efficacy. The randomized controlled trial is the means by which so much has been developed in modern medicine, and now it's time for NMO to undergo its investigation in these rigorously controlled clinical trials.